Today we will discuss the phytoplasma, but before starting with the phytoplasma, we have to know something about the mycoplasma. The mycoplasma has no cell wall. Instead of it, it has the trilaminated protein-lipid protein membrane. They has the double-stranded DNA, but in linear structure. One important point is that it can be artificially cultured. It is artificially culturable. Because of the degenerative evolution, they are different from the bacteria, and that's why they are included in the separate class molecules. And that's why they has the low size of the genomic material compared to the bacteria. Also, they are smaller than the bacteria. And how they multiply? They actually multiply by the binary fission. This is asked in many exams, that who discovered the mycoplasma? Nocard and Roux. They discovered the pleomorphic organism, which is actually obtained from the causative agent of the bovine pleuropneumonia. So they have obtained some organism which is showing the pleuromorphism. Pleuromorphism means there has no special structure, rather they change it time to time. Now we have understood that why they are changing their structure, because of the absence of the cell wall. Because they have no rigid cell wall, only trilaminar protein-lipid protein membrane, that's why they have no significant or any special shape. So now we will talk about the phytoplasma, because mycoplasma is not our concern, because mycoplasma is a broader term. The phytoplasma, Mycoplasma tesi, Acheloplasma tesi and Spiroplasma tesi. So this is the three families under the mycoplasma tails, and this Mycoplasma tesi has the mycoplasma, this Acheloplasma tesi has the acheloplasma, and this Spiroplasma tesi has the spiroplasma. But the phytoplasma is actually closely related with this acheloplasma. They have no cell wall, and they have the triple layer of the protein, lipid, protein. This is same as the mycoplasma, but the different from the other molecules of this phytoplasma is they use UGA as the stop codon. Okay, and another important problem regarding this phytoplasma is they are artificially non-culturable. The mycoplasma are artificially culturable, but this phytoplasma is artificially non-culturable. This is the problem. And another important point regarding the phytoplasma is that they don't need sterol for the growth. They don't need sterol for the growth. Phytoplasma has the smallest genome among all the living organisms. Phytoplasma has the smallest genome among the living organism. Remember, I am telling you the living organism, not the agents. Viroids, virus, those are the particles, but this is living organism. Another important point about the phytoplasma is, as they has no cell wall, that means penicillin will not work into them. That's why tetracycline is used to kill them. Now the question is, how phytoplasma is transmitted? The only source of transmission of the phytoplasma is the phloem feeding insect, and the phloem feeding insects are the leaf hopper, plant hopper and psyllids. They will eat the phloem, and in the phloem there is the phloem sieve tube, and in that phloem sieve they will enter. From that portion they will spread in the whole plant by the phloem. Talking about the important historical background of the phytoplasma is that we all know that Doi et al. First discovered the phytoplasma in the yellow dwarf of mulberry in 1971, and in 1971 EC actually discovered that the phytoplasma is actually sensitive against tetracycline, and in the 1994 under the 10th Congress of International Mycoplasma Society, decided to name this as phytoplasma. So this phytoplasma got the name phytoplasma in 1994 in the 10th Congress of the International Society. But who discovered it? Doi et al. So that's for today. That's for phytoplasma, and I will discuss the phytoplasma classification, how they infect, the important disease, how they multiply, everything in the next part.